This video demonstrates the use of the EUBrazilOpenBio platform for the computation of models of species distributions using the ecological niche modeling and its projection on geographical maps. Users who have access granted to the EUBrazilOpenBio virtual research environment will be able to reproduce this video and compute their own models and projection maps. We will compute the ecological niche modeling of Passiflora recurva, a flower present in different continents. The first step is obtaining the occurrence points where the Passiflora recurva has been found and registered. The occurrence points can be retrieved from the Species Products Discovery Service available in the portal. The EUBrazilOpenBio platform enables you to search in several databases simultaneously and seamlessly. The user will need to introduce the scientific or common name of the species they want to search for. In our case, we introduce the name of the Passiflora recurva and then click on Search. Several occurrences are found from different database sources. The user can then select them all and then view additional information from the option View Details. In this case, we have found 147 occurrence points, which can be retrieved by clicking on the appropriate tab. Results from the occurrence points can be retrieved. Those occurrence points are available on different databases such as GBIF or SpeciesLink. A variety of information is provided about the source and provider, which can give information about the quality. When all points have been downloaded, they can be saved on a file and stored on the workspace. Occurrence points are saved by clicking on Save Occurrences. The user should select the output format in order to use these points in the OpenModeler CSV and OpenModeler format. The occurrence points are stored on a file in the workspace under the name defined by the user. In our case, we use occurrences passiflora.csv. Then we can directly go to the ecological niche modeling application. The ecological niche modeling goes through 3 stages: model creation, testing, and projection. Before defining each stage, a name should be given to the experiment. In our case, Test Passiflora recurva. The first stage is model creation. The model will use the occurrence points previously downloaded. We remove previously used files and upload the one required for the experiment. Then we will select the algorithms to be used. There is a panoply of algorithms that generate a model using different techniques and which can be combined at the end. We select MaxEnt, maximum entropy, and ENFA, ecological niche factor analysis. The next step is testing. In this case, we will keep the default values. Finally, we want to get a projection of the model changing the environmental conditions. In the Projection tab, we select one layer set from WordClim. Then we submit the experiment. Once the process is completed, a file will be retrieved with the results of the projections. These files will be stored in the workspace. We retrieve one file per model. We download the files in our local disk. Files can be browsed using the proper viewer. In our case, we use Quantum GIS. Setting the appropriate color schema, we can see the outcome of the 2 algorithms. These maps represent the likelihood of finding such species in the geographical locations with the environmental conditions selected.